Welcome to the 2022 Nemmers Lecture. As most of you know, my name is Brad Sageman. I chaired the committee that selected this year's winner of the Nemmers Prize, and it's my great pleasure to welcome Kate Freeman here for a couple of reasons for me personally. And I'll tell a little bit of the story here before I ask Maggie Osborne to come up and give the formal introduction. In 1991, I started a postdoc at Penn State University, and that was the year Kate started on the faculty at Penn State. My postdoc advisor, who I consider one of the greatest mentors that I've worked with, was a fierce advocate for hiring Kate at Penn State. And from him, I kind of got this sense, this clear sense, this is one to watch. And sure enough, Kate's career has been remarkable. And when the Nemmers Prize came to our department, she was one of the first names I thought about in terms of a potential winner of the Nemmers Prize. And I was really, really pleased that the selection committee was completely unanimous in agreement on that. And so it's really been a great pleasure for me to watch this whole process. And I'm going to ask Maggie to come up now. And part of the connection here is that all the way back from '91, the type of work that Kate did inspired me so much that even though it wasn't my area of expertise, I thought it was something our department needed. And so Maggie Osborne's a member of our faculty because of Kate Freeman in a way. So I'm going to hand it off to Maggie.
All right, I wrote out words so that I didn't mess this one up, guys. As you all know, as part of the Nemmers seminar series, I've had the pleasure of introducing an amazing breadth and range of scholars associated and in honor of our Nemmers Prize winner. But I have not yet, of course, introduced the Nemmers Prize winner herself. And so it is with the greatest pleasure that I introduce Dr. Kate Freeman. The Nemmers Prize recognizes achievement and work of lasting significance in the field of earth science. And it is my job now to describe just how lasting and significant Kate's work is. You get embarrassed one more time and then you can go home and no one will talk about you again. Kate's career in earth science research started where many of ours did, and that is washing dishes in a lab. In this case, it was as a summer student at the geophysical lab in the labs of Tom Herring and Marilyn Fogel. Undergrads, take note, it's the start of a potential trajectory. After completing her degrees in geology and classical civilization at Wellesley College, Kate moved on to Indiana to complete her master's and PhD in the laboratory of John Hayes. Together, they developed a technique to measure the isotopic composition of individual organic molecules, one which revolutionized how we understand what and how biology makes organic material. And of critical note to earth science, how we observe biogeochemistry through environmental complexity. Despite being published over 30 years ago, we continue to use and gain huge insight from these techniques. As John Hayes is my academic grandfather, I can now claim Kate as my aunt. In this dynasty. After a postdoc with Stuart Wakeham at the Skidway Institute of Oceanography, where Kate got her sea legs and extended her research into marine systems, she began her professorship at Penn State, advancing through the ranks to professor, distinguished professor, and now the Evan Pugh University professor. Kate's work at Penn State focused on the application of compound-specific isotope measurements to understand different aspects of the Earth system. Her group worked to constrain the signals imparted by diverse microbes in different organic molecules in the modern system, then applied these observations to reconstruct many aspects of intervals of the Cenozoic and Paleozoic environments. Subsequently, her work came onto land, focusing on reconstructing changes in grassland, fire, altitude, aridity, canopy cover, and habitability for early humans, among others. I do not have time to describe all of the studies, as in total, they're nearing 200. But safe to say, there's hardly a biogeochemical cycle or a time period that have gone unturned. Most recently, Kate's focus has moved outward, working to understand organic matter found on Mars. Her pioneering work on site-specific isotopes as the director of the NASA Astrobiology Center for Isotopologue Research is truly at the forefront, and again, leading the field of organic geochemistry into the future. We'll hear more about this shortly. Kate has been well awarded for these groundbreaking achievements, including the Peter Schenck Award, the James Lee Wilson Medal, the Heinz Loan Stamp Science Innovation Award, the Cazarelli Prize, and the Alfred Tribes Medal. She is also a fellow of CFAR, GSA, AGU, the American Academy of Microbiology, the Geochemical Society, and I think a couple I missed, and is a member of the National Academy of Sciences. In addition to all of these honors, I do want to highlight that Kate is regarded as one of the most passionate and supportive mentors in this field. She has advised more than 30 graduate students and numerous postdoctoral scholars, many of whom we've heard from in this seminar series and who are leaders in the field in their own right. Through this network, her impact is most profound and growing all the time. And without further ado, Dr. Kate Freeman.
Thank you for those two very generous introductions. I think after that last one, I need a nap. Sorry. Wonderful. Thank you so much. And I want to just start by thanking everyone in the department for your hospitality the past few weeks while I've been in residence. It has been a true pleasure. And I want to especially thank the staff who have worked extra hard to make me feel welcome, and I appreciate that. And I also want to give a special thank you to Maggie for organizing the seminar, which we joined from Penn State in April, and I was able to come and join in person in the month of May. So thank you, it's been a treat. And as I speak today, I'll show some of the faces of the speakers who joined us over the course of the past 10 weeks. And so you may recognize a few of your new friends. So yes, I'm going to talk today about this intersection of what we can learn from the shape and properties of molecules with their isotopic signatures. And by and large, I'll focus on carbon with these words today, but of course, other elements and their isotopes are of great interest to the community and many of the people in the room here. So let's get going. And we'll start at the beginning. And that's with John Hayes. And that is the gentleman in the gray, so black and white photograph on the right. That's a picture of him in his early career years. And his former student, Dwight Matthews, is shown a little bit more mature in his years in that photo. I don't have a picture of him as a student. It was actually Matthews and Hayes that put the first gas chromatograph together with a mass spectrometer to do high precision stable isotope measurements of individual compounds. And you can see their first published chromatograms are shown on the left side of the figure. And the top one is for carbon and the lower one is for nitrogen. So John never did anything halfway. He wasn't satisfied with one element, but he got two in this initial paper. It's a really beautiful piece of work. But you may notice, those of you who work with chromatography may notice a kind of fat wobbly peaks. And that's because it was a packed column. It was a broader geometry than what we use today, and it makes wider peaks. So really my job as a graduate student was to take this approach and make them skinny and narrow using capillary methods. And that's what became the commercial product that's available and has been used, as was noted, for over 30 years. And in fact, students, just so you know, my work was not yet published, and I did not have yet my degree, and they were selling the thing I worked on already, and it was getting into the labs from the best people in the business. So I felt a little intimidated, but I decided early on that the tools don't distinguish you. It's the questions you ask and try to answer with them that's more important. So even though I was competing with some of the best labs, I tried to ask questions that others I thought would have value in. So anyway, so that's the start of things. And here's where it's gotten to. These are just a sampling of the disciplines that use this technology routinely. It's really important and fundamental to all kinds of fields in forensic applications, archaeology, lots of energy studies use it, as I'll talk about in a little bit, earth history and meteoritics. All of these fields have pieces of this. The drug test is the first item on the list, and there's this group of racing gentlemen here. And in fact, it's routinely used in drug testing. And Floyd Landis, if you heard about him, he had to give up his Tour de France medal because he was shown to have been using artificial enhancement of testosterone in his body. And that was tested or shown because what you consume and eat in the Western diet has a fair amount of C4 carbon signal in it from corn-based products. And that has a distinct isotopic signature than the source of synthetic testosterone, which is largely from soy. And so you can see in a person's blood sample that the testosterone sterol isotope value doesn't match all the other sterols that are made by the body. And you have evidence and convincing evidence of adulteration. And in fact, these instruments are now routinely in place at all major world-class sporting events, such as the Olympics. There's an isotope ratio mass spectrometer with a gas chromatograph on it to test the blood sample. So I find that kind of amazing. Anyway, all of these methods have in common the fact that the carbon isotope signature of a molecule reflects its origins and something about the biosynthesis and maybe the environment in which the organism was living. And it's those same properties that I've tried to exploit in addressing questions of my own. And we heard about some of that with the seminar series. Here's just one example of some of the molecules that we've used for geologic and paleoclimate and paleoenvironmental reconstructions. The top, on the far right side of the slide, you'll see a wiggly line that looks like a worm. That's actually a structural rendering of plant wax of N-alkane. And you can see the little blow up window shows that it's just a string of carbon atoms with hydrogens attached to it. And you can measure the carbon isotopes of those carbon atoms, or you can measure the hydrogen isotopes of those hydrogen atoms. And you can derive information about the source of through the carbon from what plant it came from, what kind of plant, and the hydrogen tells you something about the water that that plant used. And you can see in the horizontal graph on the top, that's a compilation of data of the 13C content of carbons in these plant waxes. And you see the two hump camel there. So one hump is the C4 vegetation that I mentioned that got Floyd Landis into trouble, or the lack of it. And then C3 is the hump on the left. And so these are the two major isotope groups of plants. And it just so happens that the ones on the right, the C4 plants, tend to grow in warmer, drier climates. And so there's sort of embedded in this information something about climate right there. And so we use that widely in reconstruction. It turns out that these plants sometimes burn. And when they burn, they create PAHs, which are really well-known compounds in contaminant studies and environmental studies. But they also, depending on the fuel, whatever it burns, that same isotope signal gets conveyed into the PAH itself as well. And so you can actually not only track the abundance or occurrence of fire through the abundance of these compounds, but also you can begin to discern what kind of fuel was burned to make them. And so we've used it for that purpose as well. And we heard a bit about that from two of the speakers in the seminar series, Pratika Polisar and Alison Karp, where they were really exploring the rise of C4 grasslands, excuse me, in the late Miocene and early Pliocene, starting about 10 or 8 million years ago. And you'll see in this compilation of when this sort of arrival of grassland ecosystems took place in various continental regions, was not synchronous, it was diachronous. It happened at different places at different times. And for example, in the US, what is now the United States and North American Great Plains, it happened fairly late, whereas it started much earlier in Africa and parts of Asia. And Allison has taken this sort of understanding of the occurrence of these things, and well, Pratika has taken the hydrogen isotopes to try and look at their aridity as a driver for this expansion. And then Allison has looked in particular at the association of fire compounds, the PAHs, with these different localities, or some of these localities, to really understand when they were tied to fire and when they were not. And it turns out that fire is a big driver in some of the continental regions, but not in all of them. So that's telling you something about the role of fire as an accelerant in one of what is considered by many the largest ecological transition of the Cenozoic. The other application that I've been involved in one of the other applications I've been involved in is reconstructing the amount of carbon dioxide in the atmosphere over the Cenozoic. And we heard from Yigi Zhang about his current work advancing this approach. And he was a student of my own student, Mark Pagani, actually co-advised with Mike Arthur at Penn State. And Yigi has taken the work that Mark did and sort of is pushing it forward to refine and increase understanding of this. But the basic idea is if you look at the upper sort of jagged curve on the top part of that graph, that's a temperature reconstruction by and large, and it shows kind of a drunken stagger down into the cool icehouse climates that we're in currently today. And that's matched by a downward drop in the amount of CO2 reconstructed by a number of proxy techniques. So there's a really strong association of cooling temperatures and declining CO2. And when Mark Pagani began his thesis work with us at Penn State on this problem, that association was not convincing. And so one of the really important attributes of his work was showing on a geologic timescale that if you have high CO2, you have warm climates, and low CO2, you have cooler climates. And today that's less in debate. The debate is really what do we do about human influences on that. But at the time, it was really more a question of can we demonstrate that relationship? And sure enough, there it is. So that's just an example of some of the explorations that I've been involved with at Penn State and using biomarkers to reconstruct attributes of climate, including water, carbon dioxide, biomass types, and their placement on landscape, and the relationship to climate on geologic timescale. So that's kind of a nutshell of some of that, or just some highlights from some of that work. And I'm going to turn my attention to maybe other directions now, and want to show you some of the newer things that we're working on at Penn State, and with others as well. And so we're moving from one planet, perhaps, to others or beyond. And so I just want to sort of make note of the fact that we are entering and actually have really already entered an incredible time for space exploration, and in particular related to the solar system. So planetary and planetary bodies are rapidly being sort of sampled or investigated at an increasing pace. The pace of launches of missions that are looking at the chemistry of bodies in our solar system has just jumped in the last 5-10 years. It's remarkable. And many of these missions are actually bringing material or planning to bring material back to Earth for study by Earth scientists. So we're entering really an amazing era of opportunity for studying and kind of contextualizing Earth and its inhabitants within its planetary neighborhood and the solar system in a larger sense. Here's just one example. This is a slightly older cartoon now. This is from 2014, but this is very much happening today. This is this plan to collect samples from the surface of Mars, put them in little containers, store them, and then later come and get them and bring them home for analysis. And this is really happening. There's all sorts of planning happening. The samples are already being collected and put in little bottles or little metal containers and left on the surface. And I've been involved in a couple of advisory groups. I'm sure members of the audience may also have been involved in something of this nature. One of the things I was involved in is how hot can those sample vials get before they ruin potentially biological information in the organics, right? You don't want to let them get too warm because you might, change the character of the organic material. And so there's a lot of really careful thought that's going into this, and a lot of hard work. And I think we're really on the dawn of an exciting era. Similarly, there's a mission hurtling its way back right now from sampling this asteroid. This is Bennu, and it's the OSIRIS-REx mission. And it's got in its claws and its clutches a fairly significant sample of material from the surface of this rubble, characterized as a rubble pile asteroid. And that will land, hopefully, what they say in NASA speak is a nominal landing, means it doesn't crash, in September of 2024. And so there's currently a lot of work getting ready for who's going to get the samples, what are they going to do with them, how are we going to analyze them. And I fully expect that isotopes and molecular characterization and molecular isotopes and intramolecular isotopes will all be part of that suite one way or another. So it's really an exciting time. So that brings me to sort of a topic I want to kind of touch upon, which some of the speakers touched upon as well during our seminar. And that is, when we're looking at these materials and others, there's a lot of interest in understanding the origins of those molecules from a chemical sense. How are they made? How are they potentially altered? And do they contain any information that could be considered biological in nature? So we're looking for life signatures, or maybe proto-life chemistry or prebiotic chemistry. And how will we know life if we find it, right? That's one of the big questions that's actively debated. And there are committees of people who are writing white papers and gray papers and blue papers, and I don't know what kind of papers about this topic. There's a lot happening. And I guess I would argue that we're pretty good at life detection. We've been doing it a lot the last couple of years. So some of you may recognize the COVID testing technology in front of you. Maybe some of you had unfortunate need to use this. We actually think about life detection as sort of as lay people as like, oh yeah, either it's life or it's not, right? And this sort of reinforces that idea. Either you get the dark strip or you don't. One can argue whether or not a virus is living. I think I'll leave that for the philosophers and somebody else to debate. But this idea of detecting life is it's not just for outer space. We think about it actually quite a bit on Earth as well. When we think about it for outer space material, there's actually different frameworks or sets of criteria that people debate and discuss, where it could be, rather than the strips that I showed you in the previous slide, it could be, it wiggles and, oh yeah, it's got DNA. Well, that would be pretty convincing to a skeptical audience that something's living in there. That's unlikely to be what we find. It's much more likely to be something in the pink or the yellow box, where the pink is, it's insufficient evidence of life, and there's many other explanations for it. But there's the yellow box means, okay, there's some kind of characteristic that looks lifelike, and we need to have additional supporting evidence for that. So we have multiple lines of information that are consistent. And in all of these, you'll notice that they're drawing on organic matter complexity, organic matter preference, or some combination of those two. And what I mean by preference might be the fact that in living cells, there's a lot of repeating subunits that are linked together to form a polymer. So a polymer is actually complex, but it actually has preferential structure to it as well. So it's a preference. And that preference might include conceptually a pattern of isotopes that's consistent with a living source versus a non-living source. So when we talk about complexity and preference, it can mean a variety of things to the authors of these kinds of reports. You'll notice it's organics with preference or complexity, but we already have some of that. So let me just show you what I mean by that. Before I do that, I'll show you a slide related to one of the talks that we heard during the seminar series. This is Heather Graham with her cat with laser eyes. Two cats, actually. Three cats. Yeah, it's hard to see from this angle. But yeah, so the one in the middle is Heather. And Heather did her doctorate with me some years ago on how close of a canopy were the forests of the Cretaceous. And then she joined NASA and doesn't ask that question anymore. She asks, when will we know life when we see it? And this recent paper that came out from Marshall et al. Looks at complexity as a characteristic based on how many steps, sort of reasonable ideas for organic synthesis, are required to make a particular molecule. And that's this MA, this index of how many steps. And you can see that MA more or less correlates with molecular weight up to a point, and then it kind of smears over to the right. And that's because you can have two smaller components that get put together in a single step and make a bigger molecule. So the molecular weight doubles, but it's only one step that makes that happen. So this sort of following of complexity is one idea for not knowing which kind of molecule it is, but just how hard is it to make it. And so that's an idea that's related to this notion of complexity. There is complexity, as I was starting to say, in material that's already being described on Mars. This is a former student of mine, Jen Eigenbro. She's a senior scientist now at NASA, and she's been leading the interpretation of organics signatures from some of the rover work in the SAM program, and you see that there's some evidence of complex organics in the analyses of these pyrolyzed materials that have kind of a kerogen-like structure. There's more to come. The rumor mill is replete with information about new papers following on from this work and related to it in recent papers. And they're finding hydrocarbons with different levels of, let's say, oxidation state of the carbon. So some more reduced and more oxidized carbon. So it's really exciting. So from a molecular complexity perspective, we've kind of already got it. So that's makes me wonder, how do we think about it going forward? Another approach is to look at stable isotopes in materials from outer space. And this is a recent paper from my colleague at Penn State, Chris House, where he was part of the rover team that was analyzing methane from the Martian surface. And actually, he didn't analyze it, the rover did, but he went back and reanalyzed the data to look at the patterns and saw this range from +20, which is not unusual for the Martian atmosphere, to -140, which is very much depleted in 13C relative to the atmosphere. And so he, in his really beautiful paper that sort of step by step walks through multiple hypotheses to explain this signal, and then kind of rules some out and some he can't rule out. And so at the end of the paper, he's got a couple that he sort of isn't sure about, one of which might be life, but there may be some other reasons. And this cartoon shows some of the different mechanisms that he discusses in this paper. So it's a really wonderful example of taking a signature that has a uniqueness to it, that may have potentially a life interpretation, but looking at it from multiple angles, using multiple hypotheses to try and understand its origin. So that's a nice approach to this problem. But isotopes is not a new component to our understanding of the origin of materials. And this is an older figure from Lori Leshin that summarizes some of the isotopic ranges on Mars in the red and orange, sorry, orange colors, and the green and is meteoritics, and then Earth is in the darker colors, that's actually navy blue. And one of the things that's really characteristic, I think, about life on Earth is that it's kind of a mess. And so one of the things to me is that really is a kind of a compelling life signature is the diversity of signals that we see. It's not one smoking gun, if you will. It's actually a range of properties and values. You see that in that blue bar. That's all reduced. That's the isotopic range of reduced carbon on Earth. And that wide spread of 80 per mil is directly attributable to life processes. So the signal is the noise, if you will, when it comes to life on Earth at least. On meteorites, there's a lot of organics, and some of them, the carbonaceous chondrites, is the figure we showed in our discussion of last week's paper, which shows isotopic range carbon on the horizontal axis, deuterium on the vertical axis. It shows this range of values. These are for different types of molecules. These are all based on individual molecules measured by Mark Sefton and some of his coworkers. And Neil was just telling me he was involved. Where's Neil? He's around here somewhere. There, in the back. He was involved with some of the early analyses with Dave Nemmers. And so it shows this range of isotopic range where the values of these compound classes reflect the origins of the molecules and their elements in space environments. And you can see there's some differences by organic material classes, but there's also a range within those classes. So what we're doing at Penn State in collaboration with our other colleagues and collaborating institutions is trying to kind of dig in to these individual molecules and look at the isotopes within the molecule. That requires a little bit of explanation. So let me show you a cartoon to try and illustrate this. So this is the amino acid alanine. On the left, it's shown in the form in which 99% of it occurs on Earth, which is with all the common isotope forms, carbon-12, protium instead of deuterium, nitrogen-14, oxygen-16. So 99% or more of it is in that form, but 1% of this molecule will have a rare isotope of carbon, which is shown in the right, and it can be in one of those three positions, the top, middle, or bottom shows that dark blue ball in one of the different locations. And those are all forms of alanine, just with a different isotope position. So there's two pieces of information here. One is that there's isotopologues. So that's any molecule that's the same structure, but it has an isotope substitution in it that's different than another similar structure. So all 4 structures on this screen are isotopologues. Or you can have all the same inventory of the different elements and isotopes, but just positionally they're different. And those we call isotopomers, which is like an isomer. If you're a chemist, you might know that term. And so we have isotopologues, we have isotopomers, and then if we take the whole molecule and burn it and make the measurement of the CO2 in there and the entire molecule, it would be the weighted average of all the different carbon positions. And that is what we have called for 30 years a compound-specific isotope measurement. And we're now using new language because we need to differentiate it from these position-specific measurements. And so we're starting to use the language of a molecular average. So what I've been doing for 30 years is now being called a molecular average. And that's the isotope measurement of all the atoms in a particular molecule. So that's our isotopologues and isotopomers. That's just a few examples. Here's a few more, and you can see that this isn't even all of them, right? You can have clumping on this, you can have double substitution, you can have triple substitution. It goes on, and so the structural complexity within just a single molecular form it increases exponentially with its size. And this is an area of great interest. Can we really untangle that information and have it tell us something useful about its source, the mechanisms by which it was made, and maybe the mechanisms by which it was preserved or altered. So that's the kind of work that we're interested in the center I'm directing. But this is not new, and I'm so glad Maggie made mention of my undergraduate internship, because here are those giants of the field. I actually worked with Tom Herring, who's on the right, and that's young Marilyn Fogel in the middle, and then John Hayes smiling down below. And that's Philip Abelson up in the black and white photo. And Tom and Phil Abelson worked together on a project where they clipped off the carbon dioxide or carboxyl carbon from an amino acid and then measured its carbon isotope. So they measured the whole thing, and then they just measured the carboxyl carbon. And so this is the first that I'm aware of compound-specific measurement in a biological material that included a site-specific measurement as well. And this was done actually before I was born. So this is a ways back. And so they did it all at benchtop level. This is nothing about this was automated, as you can imagine. If you're not familiar, this is an aside, if you're not familiar with Marilyn Fogel's blog, I recommend it to you. It's wonderful reading. She just recently passed away within the last month. And I do feel like a beacon has been extinguished. And so I definitely refer all of you to her blog. It's wonderful reading of a life in science, a life of a woman in science, and just a very creative and energetic soul. Okay, so that's sort of the history of it. There's actually a fair amount of work in measuring isotopologues coming from the food and flavor industry as well. They've been doing this for a while with NMR spectroscopy. This is probably the most important molecule of all of science. It's caffeine. So we're fueled by caffeine and cookies. She knows that I like my cookies. So in these studies, they actually were able to show that the ring carbons, the ones in the center of the molecule, have an isotopic value that's very different than the methyl carbons around the outside. And that reflects the biochemistry of how those methyl carbons get added. And so that's diagnostic. You can see they're trying to differentiate caffeine from green tea versus coffee. And so there's a lot of interest in using isotope information, multidimensional isotope information in flavor and food related molecules to check authenticity, right? So a lot of this comes from European companies that have very stringent rules about, if you tell me it's a strawberry flavor that's natural, it's got to come from a strawberry plant, right? And if it doesn't, then I'm not going to pay you nearly as much money as you're asking for, right? So there's a lot of money and issues of that nature tied up with it. And so they're using these position-specific isotope measurements in ways to kind of untangle authenticity of sources of food and flavor. So NMR spectroscopy has been around and it's been doing this for a while. The trouble is it takes a lot of material. So you can brew a big batch of coffee, and many of us probably do on a regular day, but and get a lot of caffeine out of it, but it's very hard to brew up a big batch of meteoritics, right? So the sample requirements for these kinds of measurements is a limitation. But the information that they have is a rich resource for us. And in fact, there's really a large body of information. This is a long ways from Tom Herring's first work. This is from a recent review article by Alexis Gilbert. He looks like a secret agent in that photograph. He's put together a really nice compilation of sort of a review of the literature of this area, showing in a variety of biochemical pathways, major biochemical pathways, position-specific fractionation, and then signatures that end up in products that we know and love as living organisms. So there's actually a fair amount of information already in place and available as we start to look at isotopologues for biological sources. So in the introduction, it was made mention that I direct a center. These are the troublemakers associated with that center. These are all the PIs. And the next set of slides, I'm going to show you some work that's come from the people who actually do all the work, the students and postdocs. And so I'll highlight their photos as I go through the presentation. But this is a collaboration between Penn State and Caltech and Lehigh and University of Texas at El Paso. And then we have our international collaborators, Alexis Gilbert and Yoshida. And now Yoshida and our collaborators at the University of Nantes. University of Nantes is where a lot of the NMR work is being done. And at LC in Tokyo, there's a lot of work where they take molecules and break them in half and then measure the isotopes of the components and then the whole molecule and compare. And so they get to site-specific measurements in a slightly different approach. I'll show you some examples of that as we go. So what I've just described in words, here's cartoon versions of. So we're really tackling isotopologues using two major techniques: NMR spectroscopy, where you expose molecules to magnets and get them all wound up, and then when they relax, they tell us something about their bond environments. And then mass spectrometry, we're actually measuring the momentum or some property of an ion that is proportional to its mass. So I'm going to share with you a couple of examples. I'm going to start with some NMR work, and we'll start with a project that is looking at amino acids and how they sorb to ice surfaces. And this was a project that was done in collaboration with the colleagues in France at the University of Nantes, where amino acids are of great interest because they're in many extraterrestrial samples, including the carbonaceous meteorites. They're also hypothesized to be in a number of these ocean or icy worlds around the solar system. So a lot of interest in them. And the question that Alison Fox was asking, Alison in the upper right, where she was wondering, how is there an isotope effect if it sticks on an ice surface? Because so much chemistry is invoked to be associated with surfaces in these extraterrestrial materials. If it sticks to the surface, is there an isotope effect? And does that tell us something that maybe is useful about understanding what gets preserved or what might be reactive? So she took ice, put it in water, and added amino acid. You can see the suites of compounds there into that water, and then let them equilibrate with the ice surface and measured. You can physically remove the ice and then measure the sorbed amino acids from that material. And look at the isotopes of what was sorbed versus the total that went into the pot. And she did this using the NMR technology. And so you can see the isotopologues, isotopomers, vertically labeled here and shown in this diagram on the right, use the NMR signals. And so you can see that the signals really differentiate the different isotopologues. And you can use the relative amounts in a quantitative sense. And that's some of the techniques that the team in France has worked out quite eloquently. And here are some of her results, which show isotope values measured for carbon as a function of carbon position, where 1 is always the carboxyl carbon in the amino acid, and then 1, 2, 3, 4, up to 7, depending on how big the molecule is. What I want you to notice is that in some cases, there's not much fractionation here in alanine, there's not very much, but in other cases at carbon 1, there's quite a bit. And so what we're really interested in is that first carbon position. That carboxyl carbon has a variety of isotopic fractionation, and they're not small. I mean, that's a 10 per mil scale, right? These are detectable and certainly easily measurable by the methods that we have. And so what is happening here is something we hypothesize is related to the non-bonding interactions and environments of of the acids in a water relative to a surface. And what we think is happening is that the molecules that are not charged and non-polar have greater fractionation at that carboxyl site because it is less influenced by the water around it. And really the strongest attractive force is between the ice surface and that carboxyl site. And this is actually not unheard of, and we see these kinds of effects in chromatography all the time. If you've ever watched a peak come out of a GC and watching its isotope ratio, you'll see it sort of goes high and then it goes low and it comes back to baseline. That's all related to these kinds of non-bonding interactions that happen to compounds during the chromatography experience. And so we speculated in this paper that that orientation is related to that amphiphilic character, or the fact that the head down is really polar and the head up, or the rest of the molecule is not polar. And it might actually have preferentially led polymerization of some amino acids relative to others. And there's some suggestion of that from other studies. Okay, so that's one. This is another example, which we actually didn't do the measurements, but we're beginning to go in this direction, which is to look at carboxylic acids in meteorites. And Neil and I were just talking about this, and he did some of the first measurements of this, which is pretty exciting. And you can see an example of the Murchison shown in the lower left, and then an example of what an organic acid, the carboxylic group, looks like in the upper figure there. And here's some of the data that's been published by Hurd et al., where they show the Murchison data and some of the Tiger Lake meteoritic data. These are all organic acids, where 2 is acetic acid, and then 3, 4, 5, 6, 7. Those are the number of carbon atoms in the structure. So they're getting bigger as you go to your right. And there's a really strong trend, if you look at Murchison data, that suggests that the first one is enriched relative to the second and the third. And if you were to extrapolate that to the carboxyl site without any alkyl carbon on it, it suggests that it's enriched in 13C. And what's interesting about this is that there are other mechanisms that are invoked for this to possibly explain it. But one possible pathway is that that carboxyl carbon itself is exchanging with inorganic carbon in a warm, wet environment. And that's something that Bob Dias, who's shown on the lower left, did his thesis on many years ago in my lab, where he measured the whole acid. Again, we see that downward trend with the hypothesizing that the one position is enriched. And then he clipped off that one position and measured it. And he showed in that upper graph, the carboxyl carbon, that it's all pretty enriched in 13C relative to the rest of the molecule, consistent with that origin. And then we did this in a variety of environments. We did it natural samples and some heated organics in a sort of a bomb to with water, with Mike Lewin. And then we actually modeled this with Jim Kubicki to show that this is an exchange reaction is quite feasible under mildly warm aqueous conditions. So it's quite possible that that meteoritic organic signal has, at least in some cases, been influenced by this kind of chemistry. Okay, so the last kind of measurements that I want to share with you, let's see how we're doing there, is with this new kind of mass spectrometer called an Orbitrap. They're actually not that new. They've been around 20 years, but they're new to isotope applications of the kind that I'm going to describe. And these are instruments that have the capability of giving us super high mass separation, so they can distinguish one mass from another at 3 or 4 decimal places. They are, turns out to be quite useful for beginning to kind of profile the isotopes within molecules. That's a cartoon of how it works. A charged packet of ions come in, and they kind of orbit, hence the name Orbitrap, around this little spindle, and they can orbit just sort of horizontally, but they can also orbit longitudinally down, up and down. And it's the longitudinal orbit that gives us the mass measurements. These are incredible instruments. They're really small. And you can see here, these are 2 examples of these instruments. This is a nickel, and that's a euro for scale. It's a little bit of a pixelated picture, but they're kind of, they can fit in your hand. And yet they can have up to 250,000 mass resolution, which is more than adequate for what we need. So they're really quite powerful. They're widely used for proteomic measurements. So if you're measuring proteins and sequences of proteins in biological studies, often it's these kinds of instruments that are being used. So what we're doing with the isotope applications of these is that you can, when we talk about separation or mass resolution, this is what I'm talking about. How far can you separate one mass from its closest neighbor? And that measurement is simply the mass divided by that distance, the small difference in mass between them. And if you want to measure isotopologues of methane and distinguish carbon-13, which is a +1 mass from its nominal cardinal mass, versus deuterium, which is also a +1, you can do so if you can make a measurement in the third decimal place, because the exact mass of deuterium is slightly different than the exact mass difference of 13C. And that, so that 0.003 divided by, or 7T divided by 0.003 gives us about 6,000. So that's the mass separation power you need to do isotopologues of methane. You want to do it for our friend alanine. Well, it's the same mass difference, but now our cardinal mass is much bigger. So the molecular mass is much bigger. And so we need more horsepower, more separation power to separate deuterium from 13C. And what's cool about the Orbitrap is that it not only has 30,000, it has 250,000 or more. And so we can easily separate these forms. And here's an example of it. This is from Kai Neubauer's work, that early paper on this. It follows up on one of the sort of the breakthrough paper by John Eiler. And he's just illustrating this. If you see in the upper left, there's a mass spectrum that shows fragment 104. That represents just clipping off the little green box and everything else. But that 104, when you add one isotope to it, would be 105. But there's lots of different forms of isotopes that can add one to it. And so you can separate C easily. Deuterium can be separated from 13C and the sulfur and nitrogen isotopes as well. So we can get a family of ion peaks, which are these different isotope forms of each fragment in a molecule. And then we can use that to begin to pull apart and measure the isotopes and then put them back together to get the distribution among the different positions. We had a speaker who shared some of her work on this. I unfortunately didn't get to hear her talk, but I know her work well, and I was really excited she was able to participate. This is Elle Shuniak, and her data are shown on the left. These are from the Murchison. So continuing our theme of thinking about organics from space, you can see that the different positions, C1, 2, and 3, have wildly different isotope values, 1, 2, and 3, with position 2 having highly enriched in 13C, reflecting a CO origin, at least that's what they interpret it as from interstellar medium. And so these origins are reflected not only in the whole molecule, but at the positions within the molecule. And that's the idea of our centers, of trying to get information like this out of different molecules. So I'm just to wrap up here, I'm just going to show you a little potpourri of some of the things we're working on at Penn State, some of my own students, and tie them to some of these ideas that I've been trying to share with you. And the first is pyruvic acid. This is a student at Penn State, Yuki Sato. Yuki has been working hard to figure out how to make measurements of the isotopologues of pyruvic acid. There's a lot of pyruvic acid in meteoritic materials, so that is one of the drivers of a fairly abundant amount of it in the Murchison, for example, but it's also a really important kind of building block in Earth's biochemistry. It turns out that he can measure a fragment of 145 and get all three positions. We can get that molecular average value. And then he can measure a fragment of 117 and get just 1 and 2. And then by difference, get the value for position 1. So that's the kind of algebra that we use to sort of unpack these different positions. This seems simple and straightforward, but it actually was really a long journey for him because he realized he had to figure out what form was actually where the atoms in the molecule are actually ending up in the fragment ions of this measurement. And it turns out that there's a fair amount of rearrangement chemistry that takes place when you ionize these kinds of molecules in a mass spectrometer. And so we actually confirm the fragment atoms, and they're tied to the original structure using labeled standards. And so that actually is a fairly meticulous work that's involved. But he just done a nice job, and now he's ready to go. This pyruvic acid is tied to the formation of acetate in modern organisms, and it's that process of decarboxylating pyruvic acid to form acetate. That's why lipids are light. It's why there's isotopic depletion of carbon in lipids. And you see that from CO2 in the atmosphere to the isotope value of the biomass. That's fractionation during photosynthesis, as it's normally described by ecologists. And then the geochemists get involved and they say, oh, the lipid value is different than the leaf value, and we call that an epsilon value. I don't know why one uses delta and one uses epsilon, but that they do, so there it is. And the epsilon value is actually vary quite a bit, but it's very characteristically around 4 per mil on the carbon scale. And this is a compilation of some really nice work that Aaron Diefendorf and his students have been doing. Aaron's one of my former students as well. So understanding why lipids are light are really at the core of all of the work that I've done in paleoclimate and paleoreconstructions. And it all comes back to this kind of chemistry where glucose is broken up into pyruvic acid, pyruvate, and then that's broken up into acetyl-CoA to form isoprenoids or the linear molecules. There's actually another pathway for making isoprenoids, which is that top one, which is characteristic of bacteria. And it's also in chloroplasts because plants took chloroplasts and made them from bacteria and made them a useful organ. And you can see that the yellow dot versus the blue and green dot differs between those two pathways. And so we're looking at that how the information in glucose is translated into pyruvic acid. That's what Yuki's working on. And then we're beginning to look at how these isoprenoids are formed and how they carry that signal from their starting material. And this is the work of Hao Ji, the postdoc at Penn State, where he's looking at isoprene and trying to distinguish that methyl carbon from the rest of the structure. And he's getting these are signals of about less than 1 per mil for these measurements. So we're moving forward on that work. And his interests are actually not biochemical, but environmental. These isoprene, as many of you may know, is emitted in large quantities by the forests of planet Earth. The global emission is on the order of 500 teragrams per year. And it gets into the atmosphere and it involves itself in all kinds of chemistry, including making aerosols, organic aerosols, and ozone, depending on the environment. It also gives the beautiful smokiness to the Smoky Mountains, Great Smoky Mountains. So I've also actually heard exoplanet people talk about isoprene because isoprenoids, it's that basic 5-carbon structure, is common to every life form on this planet. So it's actually a universal biomarker, which is kind of cool. And it ends up in the atmosphere in large quantities. So if you can see it or detect it, it might actually be a useful signature of life on a planet. I'm not sure I'm making those kinds of measurements, but it makes it kind of exciting to think about. Okay, then the last bit is just simply, I made mention that sugar has isotopic patterns that can end up in these lipids, and that we actually know that there's a fairly large isotope range just within sugar. And here's an example of that, showing the whole molecule average at 0, and the scale goes from 15 down to -10, right? So there's 25 per mil scale within a single molecule of sugar. So which atoms of that sugar get used and put into the lipid can influence the fingerprint of that resulting biomarker. And that's the idea which we're pursuing now, which is to distinguish between the methylonic acid pathway versus the MEP pathway, where we have those different atoms of glucose end up in the structure, the green dots versus the red and the blue dots. And we're just beginning, we're just scratching the surface and getting started here with this. But this is Paul Vellante, who's been looking at phycol and phytane as these very characteristic fragments that allow us to interrogate that inner atom in the chain, if you will. And maybe if we are clever and it works out, we can begin to figure out which metabolism made a molecule of the past. And so I think of this as like a very crude form of metabolomics, paleometabolomics, right? Okay. So, and one of the reasons that I'm interested in that particular molecule is thanks to the work of Kate Witowski, where she's been looking at CO2 records. I showed you CO2 records in the beginning of the talk. That ended here. And she's been able to extend that approach back in time using phytane, which occurs throughout geologic time, throughout the Phanerozoic record in petroleum and in shales and other organic rich archives. And it's done a nice job showing that its usefulness, potential usefulness as a CO2 proxy. And so our work really complements this to make sure it really is coming from a plant and not somebody else. Okay, well, you've been a great audience, very patient as I kind of revel in the nuances of biomarkers and isotopes. I'll just close with, what is it in Harry Potter? It opens at the end or something, right? And so this is ending where we started. My undergraduate thesis advisor, Tom Herring, had this scribbled in one of his books that he lent me, and it was a triangle of good, fast, and cheap, and you can pick two. You can never get all three. And I think actually with this work, it's not cheap. I wouldn't say it's very fast, but my hope is that eventually it'll be pretty good. So anyway, you have been great, and thank you very much for your attention.